Welcome everyone, and thank you so much for joining us. Our webinar today is entitled Sportomics, Metabolic Health, and Utilization of Functional Medicine Testing. And I am excited to mention our guest speaker, Dr. Jeffrey Crayer. My name is Dr. Lenora Powell. I'm a medical education specialist in Genova's Atlanta branch, and I will serve today as the moderator for the webinar. So we would like to welcome Dr. Jeffrey Crayer. Dr. Crayer, the Wellstead Health Chief Medical Officer, received his medical degree from the University of Missouri-Columbia Medical School and did his residency in adult and pediatric medicine at Indiana University, where he served as a chief resident. He completed a fellowship in musculoskeletal and sports medicine at Boston University, Boston Medical Center, and since 2011, he has practiced sports medicine in the departments of orthopedics and pediatrics at Massachusetts General Hospital. Dr. Jeff is recognized as a leading educator of patients and health professionals at Harvard Medical School and MGH. His career-long focus has been on providing the highest level of expert multi-systems care and optimizing the health and well-being of children and adults. Dr. Jeff was recently awarded his certification in functional medicine through the Institute for Functional Medicine and will shift his practice focus from specialty orthopedic to primary care. Beyond his work life, Dr. Jeff's interests includes his kids, the outdoors, yoga, cycling, fermenting, philosophy, and board games with his partner, Kirsten. In addition, you'll have access to the presentation and the slide deck on our website within a few days after the webinar. You can access these resources, previous webinar recordings, brief video modules, other materials by clicking the Clinicians tab on the homepage. I just want to quickly remind everyone that we have many non-invasive urine, saliva, blood spot, and stool collection packs that can be shipped directly to your patients for specimen collection at home, and the requisition can be completed totally online. For more information, please just visit your LiveGDX account. And so I am going to go ahead and turn over the role of presenter to Dr. Crayer.
Greetings to all attendees. Thank you to Genova for the opportunity to present this material and Dr. Powell for the kind introduction. Today will serve as a cursory introduction to Sportomics, metabolic health, and the utilization of functional medicine testing. Here you can read the objectives of the talk. The outline of today's talk includes 1, a quick definition of Sportomics, including why exercise and functional medicine are great partners. 2, a survey of the metabolome and microbiome findings of those engaged in exercise. And 3, I would like to additionally sample the literature around the application of Sportomics to exercise performance and metabolic health. Finally, I would like to intersperse some examples of functional medicine testing and its value to clinically relevant questions. Simply defined, Sportomics applies metabolomics to investigate the metabolic effects of physical exercise on individuals. Sportomics also works to advance knowledge in integrative physiology and the systems of biology of movement, with a goal to translate markers associated with metabolic challenges of training or competition to the similar stresses of disease settings. As technology advances and continues to become less expensive, it will continue to mature Sportomics from its infancy of metabolome and microbiome investigations further into the more mature and enlightening investigation of the epigenome, transcriptome, and proteome. Despite my sports medicine specialty, I personally find exercise and movement to be the second best medicine we have available, behind food. But it may be the easier of the two to study. Exercise is medicine, and a medicine with a great benefit to risk profile. In addition, exercise provides clinically relevant questions that I find rewarding, such as, what is the tipping point of this powerful medicine? Who are the responders? Why do they respond? And what can we do to help those who do not respond to physical activity and movement? Exercise is a lifestyle intervention within the biopsychosocial model of medicine. it stresses systems biology, thereby offering us insights into how our metabolism and our bodies work and how to maximize health and function. Exercise is mitochondrial medicine with pervasive effects on gut health, immune health, metabolic health, brain, nerve, and mental health. Sportomics is multivariate with major variation in the intervention, whether it be the mode, the frequency, intensity, or time, major variations in subjects, whether they be male or female, young or old, and all with their own individual genetic predisposition. Take that and the additional layer of many confounding variables such as nutrition variability, training history, and the level of health or disease, and you have quite an interesting puzzle to work. All variables then act on the system biology with crosstalk from the cellular function through the organ function to whole body homeostasis, all toward what I consider to be the billion-dollar question. What is the proper hormetic dose of movement? Sportomics does have many challenges, and many are trying to move beyond correlations and associations. Based on the success of the science paper by Zevi et al. Out of Israel, who worked with machine learning around predicting glycemic response to food based on functional medicine testing, demographics, questionnaires, and food intake, some are applying similar methods to exercise, which we will revisit later. Can we use functional medicine testing to predict performance measures such as VO2 max, or equally important, the glycemic and inflammatory response to exercise? All this in the effort to get closer to the answer of a personalized hormetic dose of exercise. So what does the Sportomics literature say? A summary of the metabolome investigations points to substantial heterogeneity of the studies. The drivers of metabolome change being exercise volume, while metabolite changes being short-lived, there are obvious changes between consistent movers and inactive patients. Generally speaking, there are lower amino acids, especially branch-chain amino acids, due to increased gluconeogenesis, increased lipolysis, and increased beta oxidation of fats. However, as this image depicts, even the modes of exercise change the findings. These differences are likely due to the variability in the exposome, or the xenobiotic profile of different sports. For example, pesticide and fertilizer exposure in grass sports, plastics exposure in turf sports, potential chlorine and bromide exposure in pool sports, along with different diets, supplement, and drug associations with each individual sport. Here you can see a graphical representation of the response to acute exercise, with most effects on metabolites lasting minutes to hours. But generally speaking, there's an increased lactate and pyruvate from glycolysis, separate from the markers of beta oxidation and a use of ketone bodies. Hinted at the lower half of this image are additional correlations of impaired exercise pathways in those with metabolic disease and insulin resistance. Separately, and not shown here, the researchers found metabolome perturbations and prediction of VO2 max around a specific inflammatory signal at 15 minutes after exercise. So what about our consistent movers? Consistent movement, or athletes, however you want to define them, is associated with a healthier metabolic profile and a healthier metabolome. Again, we see the lower amino acids and branch chain amino acids, but what really stands out is the changes in several lipid metabolites. This image is simply to highlight the need and current direction within Sportomics to investigate the epigenetic modifications in response to exercise. This includes changes in mitochondrial function, but more importantly, the 1-carbon metabolism along with histone and DNA modifications through methylation, demethylation, acetylation, etc. The goal being to look at the proteome and the transcriptome to see what changes the response to exercise over a chronic duration. So here we have metabolomics result of a patient of mine shortly after the pandemic started. She is a 38-year-old high-level athlete with a normal BMI on a pescatarian diet, seeing me for brain fog. She was concerned and anxious about her broadly low amino acid markers, but I was able to allay her concerns that it was more a sign of her great aerobic fitness due to the increased use of branched chain amino acids for gluconeogenesis and lower level of amino acids that are associated with insulin resistance. Moving on to the microbiome of athletes, Clark et al. Was one of the first descriptors of the microbiome of athletes. This was in 2014, and interestingly, the group he chose to investigate were rugby players, a relatively high BMI group, but they were able to use both a low and high BMI inactive control group. Clark et al. Were the first to describe the higher proportions of Akkermansia muciniphila in athletes, but also those being found in lower amounts in the higher levels of Akkermansia, both in the athletes and the lower BMI controls, not in the high BMI controls. In addition, they showed a greater microbiome diversity in the athletes, and unfortunately, their findings were complicated by a much higher protein intake in the rugby players. So that set off a wealth of microbiome studies, and with follow-up studies confirming the greater diversity of the athlete's microbiome, but expanded upon the findings to include higher levels of short-chain fatty acids in the athletes. This brings us to another great question in Sportomics. Is exercise good for the gut or bad for the gut? As Patty Devers and Michael Chapman say on Genova's podcast, The Lab Report, it depends. Exercise generally increases transit time, diversity, butyrate, and short-chain fatty acids, but it can also increase intestinal permeability and metabolic endotoxemia. A 2021 article in the Frontiers of Nutrition reviewed the literature and tried to answer this question of a tipping point for increased intestinal permeability. They did use metabolic endotoxemia as their outcome measure, mostly defined as inflammatory markers and LPS markers, or lipopolysaccharide markers. And they did seem to increase in the blood at 60 minutes of vigorous training at 70% of VO2 max. Some of you may be saying, well, what is that? 70% of VO2 max is the upper limit of zone 2. What is zone 2? Zone 2 is a level of exertion that passes the talk test. You can easily hold a conversation. However, this increased intestinal permeability is also altered by other variables, such as altitude, environmental temperature, fluid restriction, age, and even trainability. This is where we might look to some of the research around the heat shock proteins. How do we limit the changes in intestinal permeability? Well, the answer to that, as you can see from this image, is a healthy mucin layer. So as Dr. Robert Lustig has been known to say, feed the gut bacteria so they don't feed on us or our mucin layer. Hence, we now see why the increase in the short chain fatty acids is so important. Nourishing this diverse microbiome with fiber, both soluble and insoluble, provides that protection and precursors for the increase in the beneficial effect of short chain fatty acids. The mechanism in which the short chain fatty acids act on endurance performance are still being investigated, but we do know that there are receptors on skeletal muscles separate from the small short chain fatty acids' ability to increase colonic gluconeogenesis, in addition to their ability to work through the gut-brain axis, and even potentially acting as 2,3,4 carbon metabolites for the TCA cycle and energy generation. Here we see the feed-forward cycle of microbiome health through exercise and sport. The benefits at the bottom of higher microbial richness, higher abundancy of beneficial Akkermansia, Veillonella, which we'll come back to, and Prevotella, along with this selective advantage for lactate utilizing bacteria. Lactate, something we'll come back to too. The benefits being that increased production of short chain fatty acids, neurotransmitters, maintenance of the intestinal barrier function if we are not overdoing it or causing excess inflammation, a beneficial effect on the immune system, and improved energy harvest and utilization. This is a busy slide, so I'll give you a little time to look at it. I don't want to spend too much time on this image because I'm not dogmatic around any food plan, including plant only or carnivore, but protein and carbohydrates have very distinct effects on the microbiome and very distinct effects in athletes. High intake of protein without fiber leads to increased proteolytic microbes and their derived end products that can negatively impact intestinal permeability and cardiometabolic health. However, with fiber, there is suppression of protein fermentation by controlling gut pH. And this is where I agree with Dr. Kalish in the name of cardiometabolic health. Test, not guess, and I suspect the future will include more regular monitoring of TMAO with the popularity of the ketogenic and/or more protein-forward diets. We do not know we do know that high fiber promotes Prevotella growth and increases short-chain fatty acids, but also low fiber promotes the growth of mucus-degrading bacteria. The complicating factor is fiber is not necessarily tolerated by all endurance athletes or even our common patients with gut dysbiosis. So what about Sportomics, the microbiome, and exercise performance? I want to return to that question of lactate producing bacteria and the implications for exercise performance. This really impressive study done in 2019 by Scheiman et al. Sampled the stool from 2015 Boston Marathon runners, both pre and post marathon. And they found increased Veillonella species. Now, why do they care about Veillonella? The heat graph up on the top shows you that Veillonella species contain all the enzymes to thoroughly metabolize lactate into propionate. So endurance exercise is a high lactate environment for energy and metabolic reasons. And you can expect to hear about the lactate shuttle in any talk covering energy and exercise physiology. In addition, they found that when they took a gavage of the human Veillonella atypica from the humans and put it into mice, there was an increase in the treadmill run time. But why? Searching for the biological plausibility and testing the hypothesis that the microbiome was involved in exercise performance, the group settled on propionate. One, because of the metabolic pathway, but two, because they took separate step of infusing propionate into the mice and seeing the same increase in treadmill run time. This is just a restatement of what I just said. To confirm that potential mechanism, they did infuse the propionate into mice without a bacterial gavage and found the same increase in performance. This spurred a mini buzz about fecal transplants to increase performance, but don't worry, this hasn't been done in humans, at least to my knowledge. Interesting, getting back to the systems biology of exercise and the non-reductive nature of functional medicine, Ludberg et al. Proposed another potential mechanism of action, given their knowledge that Veillonella species are nitrate reducing in the oral cavity. This leading to increased nitric oxide and improving both mitochondrial and muscle performance. So is it A, B, or A and B? We don't really know. So can one apply these findings to functional medicine testing? Here you see the lactate producers in green, the lactate degraders in yellow. And this is one of the many patterns that I try to put into my mind when looking at the GI effects. But what you'll see is they're both degraders and producers within the firmicutes. And in my opinion, meaning not scientific fact, I suspect much of the contradictory literature around the FB ratio revolves around the mixed performance of the firmicutes phylum. Again, no scientific fact, just random musing there. I can only add anecdotally, armed with the above knowledge, that I'm seeing a lot of this pattern in my patients with issues around mitochondrial function, whether it be pain, fatigue, brain fog. You can see here the GI effects of one of my patients with chronic fatigue syndrome, separate pain amplification syndrome, and exercise intolerance. She had previously failed a graded exercise program. But with that knowledge, I can look at this and educate her that yes, she is very deficient on short chain fatty acids, and she has very low levels of the Rosaburia, Ruminococcus, and Veillonella species that can help with lactate metabolism. If you're really astute, you'll also see a paucity of the bacteria globally and also with the lactate producers in this one case example. So moving from the microbiome and onto sportomics and metabolic health. As we know, cardiometabolic disease is one of the top, if not top, cause of morbidity and mortality in developing countries, developed countries, I apologize. However, only 12% of the adult American population is metabolically healthy. Again, 88% of the adult American populace is not metabolically healthy. While most of us in functional medicine are preventative minded, if you're not addressing nearly everyone's metabolic health, you may be doing them a great disservice. In addition, as a pediatrician, I must say, as our current pediatric and adolescent population ages, I'm not certain that the metabolically healthy 12% of the population will move in a favorable direction. So I like this image because it shows the Cori cycle or the lactate shuttle. In addition, it has the image showing lactate working in the gut microbiome. It's no longer just the liver and the muscle. It now involves the microbiome. And a couple of buzzwords in the metabolic health and fitness community includes metabolic flexibility. This is the ability to adjust your metabolism to break down the macronutrient present. While I am no friend to excess fructose or glucose, although I won't admit my sweet tooth, in my humble opinion, having your health wrecked by a sugar molecule is not a state of optimal health either. While I never promote excess fructose or glucose, the inability to metabolize sugar is not necessarily a state of optimal health. Again, one must hear about lactate and the lactate shuttle if we're going to address this metabolic flexibility and/or metabolic health. Lactate has gotten a bad rap, mostly at the hands of the fitness community. However, we as medical professionals and experts in cellular energy can now re-educate the populace about the great benefits of lactate. If you can take one point from this lecture, I hope it's this one. Lactate is the universal fuel. Lactate supplies acetyl-CoA to working muscle and competes with glucose and fat for oxidation. And within the cell, it's oxidized to pyruvate for access to the mitochondria and to offer more protons to the NAD for energy generation. In addition, lactate inhibits lipolysis in adipocytes, and another reason why you might want to stay in the oft maligned, quote, fat burning zone. I love my high performance athletes, but they all need a little bit more of zone 2. Think talking test. So think about this. What's the PET scan? It's radio-labeled glucose. What do you see light up on a PET scan? The brain, inflammation, and tumors. If you do this similar study and take radio-labeled lactate, you see a greater increase in radio-labeled intermediates of the TCA cycle. There's more lactate being funneled into the TCA cycle than there is glucose. So what goes wrong in metabolic disease? And I apologize, I did not define this early on. To me, metabolic disease is insulin resistance. I don't believe in such a thing as prediabetes, you're not pregnant or not pregnant, all those wonderful cliches we like to throw around. But when is the body tipping into insulin resistance or metabolic dis-ease? We know those with excess weight or visceral adipose tissue walk around at higher baseline lactate levels. They also have a relatively or absolutely higher baseline glucose level. So some have labeled this poor metabolic condition as what you see there, the vicious Cori cycle. Cori cycle being for the pair labeling the lactate shuttle, in which cells are starved of energy due to impairment in the Krebs cycle or the TCA function. So even worse, in the state of poor metabolic health, the muscles are demanding energy and getting the limited ATP generation from anaerobic metabolism because the TCA cycle is dysfunctional, while the liver has to invest ATP to generate more glucose for the demanding muscles. So stop starving the liver, stop demanding so much of an energetic investment of 6 ATP to change the pyruvate or lactate back to glucose so that you can feed your muscles. The liver does so much for us, yet we want to fill it with fat and starve it energetically. No wonder it struggles to keep us with all our other demands, struggles to help us with all the other demands we place on it. So here's a nice flow diagram on the cascade of dysfunction leading to metabolic disease. You have at the top there the reduced mitochondrial mass and function, leading to reduced aerobic oxidation. You have a compensatory increase in anaerobic glycolysis, which gives you metabolic inflexibility and metabolic disease. So what's missing there is a circle that goes back up to metabolic disease, further promoting more mitochondrial dysfunction. So what does the vicious, quote, vicious Cori cycle look like on NutrEval testing? And the following pattern may stick out to you, or I encourage you to start looking at your urine organic acid testing for this pattern. Now, these are relative changes. I will not say that you are going to see a bunch of elevated yellow-red lactic acid, but you'll see it on the higher level of normal, especially in relation to where pyruvic acid sits within its normal range. In addition, you may see low citric acid, often seen more with severe obesity, and then the lower TCA intermediates, labeled by some as the hypometabolic state or mitochondrial retraction. Mitochondrial retraction is metabolic disease. And then you may or may not see higher malic and succinic acid. So we come full circle. Who responds to exercise and who does not? Why are there reports of 7 to 70% of patients not responding to exercise in terms of insulin sensitivity and glucose homeostasis? Dysbiosis leads to insulin resistance through increased intestinal permeability, metabolic endotoxemia, changes in the small chain fatty acids and branch chain amino acids, and perturbations in bile acid metabolism, which I haven't even mentioned today. Many have heard about the 2017 study of fecal transplant improving gut diversity and glycemic control when taken from a metabolically healthy adult into someone with metabolic disease. So Liu et al. Out of China published a paper in Cell Metabolism looking at the question of who improved metabolic health in response to exercise. So following the lead of the previous image you saw of Zevi et al. In the application of machine learning and analysis to a clinically relevant question, they defined the responders as those that showed a decrease in the HOMA-IR, the homeostatic measure of insulin resistance, to greater than 2 times the technical error. HOMA-IR is not the perfect measure, but one of the most easily accessible with a fasting insulin and fasting glucose. Interestingly, they did not find changes in the alpha or beta diversity both pre and post exercise. However, Bacteroides genus and Clostridial order varied post exercise, both of which are involved in short chain fatty acid production. In addition, butyrate producing firmicutes was promoted. Oh my, where to start? I just love this image. And if there is one image to add to your knowledge bank in assessing metabolic health through functional medicine testing, it would be this image. What you see here in front of you are orange dots that are metabolites associated with insulin resistance. And you have blue dots, which are associated with insulin sensitivity. The lines, the blue labels and the red labels are those that are upregulated in responders in blue or upregulated in non-responders in red, with the one exception up in the right side where it was attenuated in non-responders in the production of butyrate. So take a moment, find in this image the responders. What did they do? They increased their butyrate production. They decreased their branch chain amino acids. They decreased separately, not shown here, their branch chain amino acid producer, or Prevotella. They increased the catabolism of branch chain amino acids. They increased the production of propionate and the biosynthetic enzymes associated with propionate. What did the non-responders do? They took the abundance of aromatic amino acids and increased the phenolic derivatives of aromatic amino acids. They increased the sulfate from the sulfur-containing amino acids, and they increased the production of glutamate without the continuation to arginine, good for nitric oxide health, and into GABA, again, keeping things at arginine and attenuating the production of GABA. So what does that look like on a NutriVal? I told you I love patterns, and this is another one. Worry less about the absolute results that you see on this image and focus on the areas, the arrows, sorry. Unfortunately, I see too much of this pattern and not enough of the following pattern, which is much simpler. You're seeing here the increase in the branch chain amino acids, valine, isoleucine, and leucine, the sulfur container and aromatic amino acids, along with the increase in glutamic acid and decrease in GABA. Again, this is relatively speaking. We are not talking about really high beneficial GABA, but more on the higher limit or higher end of the normal range. So what does the more beneficial pattern look like? A decrease in the branch chain amino acids, an increase in arginine and histidine, along with higher in the normal range of GABA. Lucky you, this is my concluding slide. And I'd like to focus on the bottom here. You can read the associations found on functional medicine testing above, but below you'll find the action items to promote a better metabolic health. Success is the name of the game, and like any good parent, we must pick and choose our battles. If someone has not responded to exercise, stop blaming and shaming. Acknowledge the fact of non-responders. And I apologize for not including this previously, but non-responders to exercise is 70% or 2/3 of the population. So while exercise is a great medicine, we need to work to help our patients get the full benefit. So how do we do that? How do we change, acknowledge the fact of the non-responders, and how do we change the physiology from a non-responder to a responder? And I hope it's clear that we have to work on the gut health. Feed your gut with fiber, resistant starches, fermented foods or probiotics, and consideration of kickstarting things with some butyrate. Again, that doesn't necessarily get to the root cause, but to get buy-in and momentum in the right direction. Sometimes you may have to supplement. Separately, advise a lower exercise intensity, but continue to work on increasing volume. Trust me, I love my performance athletes. I love those playing with the edges of exercise physiology, but simply stated, for metabolic health, you need to shoot for zone 2 exercise or movement, which passes the talk test. So we finally reached the end. In conclusion, Sportomics has identified changes in the microbiome, in the metabolome of athletes that are relevant to metabolic health. The future will include deeper analyses into the omics of exercise. But until then, you can use the previous summaries of functional medicine testing findings to help guide current patient care, educating patients of the desired changes in their lab tests, the reasons prior attempts of using exercise as medicine have failed, and how to improve on that outcome in the future. And with that, I'll respectfully turn it back over to Dr. Powell.
I am in awe. I am in absolute amazement. Where were you when I was actually an NCAA athlete? I felt this information was extremely valuable, extremely wonderful. It was just a great presentation. I have like 2-page worth of notes, but now we are going to kind of transition a little bit into a few different questions that we did receive. So there's a question about how much does the microbiome actually fluctuate during exercise? Because we know it fluctuates quite a bit. with your diet. And we do see in literature that exercise can impact the microbiome, but by how much?
That's a great question. The question being, what is the effect size of exercise on the microbiome? The literature will say it is all across the board, including some of the most recent studies cited where there were responders and non-responders. There was not a major change in the diversity of the gut. So we are looking at patterns and associations. But to get to the question's point, yes, the microbiome is a very dynamic microenvironment, and exercise does change it, but so does the break from exercise. So that is that question of beta diversity. And if you really want to pin the scientists down on this without multiple samples, you are catching a one-time snapshot, and we have to interpret all of these functional medicine tests in light of what were you eating at the time, what were you supplementing at the time, what were you exercising at the time. So generally, most of the studies show microbiome changes with exercise and a resolution with rest. The timing of those changes, I don't want to miscommunicate. I don't know that I have that answer necessarily, but it does go back with the training, so to speak.
No, I think that's great. Just really pointing out the many factors that influence the microbiome. But I think you brought up a really another wonderful point, which is when you are not exercising or when you are resting, there also can be another shift of the microbiome. So in regards to specifically Veillonella and Akkermansia, I wonder if patients who naturally have this ability to perform at a higher level, if they tend to just have higher levels at baseline, or is that not a fair thing to say because it might vary from patient to patient and depending on their activity level?
I think it is fair to say, and most of the literature, when you dig deeper, will say the adolescent, early adulthood is a great conditioning time. So the metabolomic and microbiome changes do have a home residence that seems to represent more of early life activity or inactivity than it does what you're doing at the time. Yes, you can do acute changes and see those changes. So I'm not sure that I answered the question specifically, but I just wanted to come back to the previous question that investment at an early age or lack of investment at an early age makes the changes easier. So with Veillonella and Akkermansia, we definitely can see, to my knowledge, changes in Akkermansia related to a gut restoration program or things like that. That's one of the few studies showing change. Of course, industry sponsored, but it is about the only thing we have to hang our hat on for interventions in the microbiome. Do the levels of Akkermansia and Veillonella, I think, represent more the lifelong exercise history rather than what you're doing at that moment? Yes. Do acute changes in your exercise change those levels? Yes. So I'm speaking out of both sides of my mouth, and I can admit that.
No, I think, again, that's wonderful. I love it. I like the fact that you pointed out that there is an influence earlier on in life and how your microbiome reacts later on in life, but there is that acute change that can happen. So how can we use lactate levels when looking at organic acid testing in assessing a patient's metabolic flexibility? Like, can we say that high levels of lactate are good, or is that kind of simplifying it a little bit too much?
No, I don't have an immediate answer to that. I can tell you, as I've dug into the literature, I went through all my prior testing on my patients, and many times the lactate and pyruvate are within normal limits. But what I did see, again, no scientific proof of this, is where are they in the normal range? Is pyruvate generally low and lactate on the higher end of normal? I'm not seeing a lot of outside normal ranges, so to speak, because as I kind of alluded to, 88% of even the baseline for standards in Genova testing or anyone's testing is not necessarily metabolically healthy. So I can't answer the question directly. I can say, looking and through my few years of testing with Genova, it's about the relative ratios, pyruvate to lactate, and I do like the GABA to glutamic acid. Just where are they in relation to each other? That did seem to correlate well with whether that patient was metabolically healthier or metabolically unhealthy. There's no one lactate level. There's more research in the serum lactate level than there is around urine organic acids.
Understand. So with the case that you brought up, the 40-year-old patient, female patient that had, you mentioned mitochondrial challenges. I believe you mentioned she had chronic pain and some fatigue and some exercise intolerance issues as well. When you showed her results, we did see very clearly that her short chain fatty acids trended low, as well as her microbiome overall trended low, including some of those important bugs that you did point out. So for that patient in particular, what would be your starting recommendations to help shift the short chain fatty acids and the microbiome as well?
Yeah, so we are actively working together, and she's also a perfect example of your patient who is somewhat intolerant of fiber. So specifically, this is where I jump beyond root cause and try to kickstart things with some butyrate or something to get a little more short chain fatty acids into the system to just skip the microbiome. And we have a very slow progression of soluble and insoluble fiber. So I actually have a ramping program for fiber for those that are invested in it and understand the value. So we are currently going through that process of building tolerance to fiber, because that gets more to the root cause, and feed the bacteria that are going to be beneficial for us and provide or make the beneficial metabolite of short chain fatty acids. So specifically, while supporting the mitochondria is good, that's part of the program too, supporting all mitochondrial function. We did go at the gut very hard because it's where her biggest pain point was too.
And for that patient, you mentioned that for people who tend to be non-responders, that you have them start out on an exercise program that's not so rigorous. What would be a potential consideration, like walking several times a week or doing more cardio several times a week? What would be more of an exercise recommendation for that specific patient who seems to be intolerant for exercising?
So exercise prescription is driven by the patient. I generally lean towards endurance exercise or something zone 2 that's more for the mitochondrial health, and that would be walking. However, if walking leads to more pain or it's just not something the patient will do, then there's I love resistance exercise because generally those with a little more mass can move a little more mass and feel very positive about the gains made in resistive training. And muscles are our #1 sink for glucose. So whether it's an endurance or resistance prescription, or both, but more often picking one to start with, it's based on what's tolerable and personalized to that patient. If we can do the endurance, it is simple walk or stationary cycling if they need to take the excess weight off their joints, and focusing first on volume, keeping it ridiculously simple, getting the goal up to 150 minutes, and I will divide that amongst 3 days a week so that there is recovery time. And once we can build from 45 minutes divided amongst those 3 days, then I will try to push the 45 minutes up to 60 minutes. So volume before increasing frequency, because I find that the patients that struggle getting started also need more time for recovery. So while there's value in going from 3 to 5 days or increasing frequency, I'd rather increase the volume first, then the frequency. Again, just building momentum. And when I talk about building momentum, this is over 8 to 12 weeks. I do not have any excessive goal before 8 weeks. It is ridiculously simple. And if that is starting once a week, it's starting 1 day a week, split up amongst the day, trying to get to 45 minutes. The value in going longer is probably a higher return on the investment than going more frequent. However, they are walking through the day. So
I really like that. Another exercise that I sometimes like to think about is swimming, especially with people who may have just joint aches and pains, because it can help alleviate some of that pressure from the joints. But I love what you said with regard to the volume, not necessarily the frequency. And I think throughout your presentation, you proved that very clearly in regards to how there is benefit in the resting period. So can you talk a little bit more about impaired pyruvate oxidation, or this vicious Cori cycle? I know you mentioned that we typically will see lactic acid a little bit on the higher side, the citric acid cycle intermediates a little bit on the lower side, as well as the lower trending citric acid. You know, sometimes when I see that pattern personally, especially the lower TCA intermediates, I wonder about amino acids. Can you talk about that just a little bit more? How do you approach that particular vicious Cori cycle?
So I don't have a specific approach. Branched chain amino acids is a push button item in a lot of my resistance athletes. And I don't want anyone to walk away saying branched chain amino acids are bad. They are good. It's good when your body can metabolize them. So you are spot on, Dr. Powell, that branched chain amino acids are degraded into some of those intermediates or help feed the cycle. The one concern is it's always what is your body doing with it. And in a setting of poor metabolic health, there is a theoretical concern that the branched chain amino acids are not being metabolized and regenerating the TCA intermediates. Now, that's all theoretical, so I can't, this is, you know, just guessing as to which way things are going, but I'm not opposed to trying branched chain amino acids, and there's an obvious reason to think they could be helpful, but they're also associated with insulin resistance. And there might be a reason that the resistance or the bodybuilders like them, because you don't build mass, generally speaking, without insulin. So if you want to make that connection too, maybe the branched chain amino acids in a less metabolically healthy patient can also lead to increased mass accrual good if it's muscle, not so good if it's fat.
I just want to play on this hypothesis or this theory because I think it's so incredibly fascinating. It kind of makes me wonder if potentially those who have a need for amino acids, if they have better short chain fatty acids, if they would be able to operate a little bit more effectively than individuals who have low amino acids along with low short chain fatty acids, and say, you know, let's put on top of that a poor microbiome health. So it's almost like you wonder if supporting the microbiome, short chain fatty acids would be that extra layer of benefit for amino acid therapies.
Very well stated, and potentially, I agree. I have no hesitation to branch chain amino acids in someone who looks like they are metabolically healthy, insulin sensitive, and could you spur that state or foster a more metabolically healthy by helping short chain fatty acid production? Absolutely makes sense to me. If I understood you correctly, I think I did.
Yeah. I like to hypothesize. So when it comes to fatty acid metabolism, how does that play a role, if any, into this whole entire story?
So fatty acid metabolism is a whole nother talk or two. This isn't directly your question. There is something called the athlete's paradox. And if you look at the muscle fat content of an athlete and someone with type 2 diabetes and obesity, they look very similar for lipid content. So an athlete's muscle actually stores energy within the muscle as glycogen and as fat or lipids. That energy is metabolically bioavailable to someone in good metabolic health, whereas you see the similar appearance in the muscle of someone who is metabolically unhealthy. So there are no shortage of studies around high-fat diet and the ability to increase beta oxidation in response to high-fat intake. And what we can say shortly, everyone defines high-fat intake differently in the literature, but those that are metabolically healthy do increase beta oxidation in response to fat. Those that are less metabolically healthy do not increase beta oxidation to match the fat intake. So it's an extremely interesting aspect of metabolic health that I didn't even touch on in this presentation, other than when you look at the there was the one image where the chronic exercisers had metabolome changes around lipid intermediates. And so it's not even lipids and free fatty acids, but the ratio of triacylglycerides to diacylglycerides within the myocyte lipid content. Sorry, that was a very long-winded answer.
No, I think you popped a few of my brain cells, but it's okay. This is great because now I'm starting to think when I look at metabolomics testing or the citric acid cycle, and I'm looking for how we are metabolizing our fats with regard to the subearic and adipic acid, we really can't just assume give, you know, certain nutrients to help move those fats into the mitochondria more effectively. We have to look at the whole picture of metabolic health, which will then lead you back to the microbiome, which then leads you back to the short chain fatty acids. I just think that this story is just so fascinating and how you really built it with regards to Sportomics and how you really broke it down with showing us those examples of functional medicine testing. And so I apologize apologize because it is at the top of the hour and we will have to end our question and answer period there. So I just want to remind everybody who's still with us, additional educational materials, you can visit our website at gdx.net. You can call client services. They will be able to support you. We have complimentary appointments with our medical education specialists to answer any question that's related to our testing, including choosing the right test and reviewing patients' test results. Also, please check out our website, gdx.net, for upcoming webinars. We also have the podcast that Dr. Creer mentioned, the lab report. It focuses on functional integrative medicine, specialty laboratory diagnostics, natural therapeutics, hosted by Drs. Patty and Michael, and an assortment of guests. So I hope you all enjoy this presentation as much as I did. I just want to, again, from the bottom of my heart, Dr. Creer, I just want to really thank you for such a great presentation. I think you just really, you challenged a lot of our assumptions we made with regard to people who are considered to be non-responders in terms of exercisers. And I really appreciate you for doing that. I think it's great for us all to think.
My pleasure. Thank you so much for inviting me.
Awesome. Well, everybody have a wonderful day. Goodbye now.